Your DNA is a master blueprint containing the exact instructions for every single protein that keeps you alive. But there is a logistical problem. That blueprint is trapped inside the nucleus, the cell's heavily armored vault, while the protein-building ribosomes are floating far outside in the cytoplasm. The cell cannot risk sending the original double-stranded DNA out into that turbulent environment. If the master code were to be damaged or degraded in the cytoplasm, the loss of information would be permanent. To bridge this gap, the cell uses transcription. This process locates one specific genetic instruction within the archive, opens the helix, and extracts a temporary mobile copy that can safely leave the vault. Without this copying process, the information in your DNA remains a static archive. Transcription is the link that turns those dormant instructions into the active messages that actually build your body. The engine behind this process is an enzyme called RNA polymerase. It's a powerful molecular copying machine, but it's also completely blind. It cannot find the start of a gene on its own. To find its place, the polymerase relies on molecular scouts called transcription factors. These proteins scan the genome to locate specific landing sequences upstream of the target gene, known as promoters. This map of a transcription unit shows the landing pad in detail. The promoter contains regions like the TATA box, roughly 10 base pairs upstream, which anchor the transcription factors. This guides the RNA polymerase to the +1 start site. This strict initiation checkpoint acts as the cell's gatekeeper. By requiring these precise coordinates, the cell avoids wasting energy on unused instructions and ensures that only the relevant proteins for a specific task are manufactured at any given time. Once docked, the polymerase pries open the DNA double helix, exposing the individual rungs of the genetic ladder. As the machine moves, it only reads one side of the DNA, the template strand. According to biological convention, the polymerase reads this template in a strict 3' to 5' direction. Using free-floating nucleotides, the polymerase assembles a brand new molecule of messenger RNA in the opposite 5' to 3' direction. It builds this sequence using simple chemical base pairing, matching every C on the template with a G and every A with a U. When you compare this new mRNA sequence to the unused coding strand on top, the relationship is clear. Because of how the bases pair, the mRNA is an exact replica of that top DNA strand, only with uracils substituted for thymine. This process, fueled by energy from ATP and GTP, continues at high speed until the polymerase hits a terminator sequence and detaches from the DNA. By reading the DNA asymmetrically, the cell creates a chemically distinct, disposable copy of the code. The master DNA strand then snaps back together, completely unaltered. In simple bacteria, this raw RNA is immediately ready for use. But in complex human cells, the raw transcript is fragmented and requires significant processing before it's functional. This schematic of human DNA shows the problem. The valuable instructions, the exons, are interrupted by long stretches of non-coding junk called introns. At this stage, the molecule is just a rough draft, called pre-mRNA. To prepare this draft, the cell adds protective armor, a cap to the 5' end, and a poly-A tail to the 3' end. Next, the spliceosome cuts out the useless introns, seen here as discarded loops, and glues the remaining exons into one readable message. This editing also allows for alternative splicing. From the same raw transcript, the cell can choose to connect exons 1, 2, and 4 to make one protein, or swap in exon 3 to create a different variation. This intensive editing is a primary driver of biological complexity. It allows human cells to extract immense versatility and multiple different tools from a single gene. Fully processed and mature, the mRNA finally exits the protective vault of the nucleus and enters the active cytoplasm. Here, it meets the ribosomes, massive factories made of ribosomal RNA. They clamp onto the transcript and begin translating the digital code into a physical product. As the ribosome reads the code, transfer RNA molecules arrive. As shown here, the tRNA uses an anticodon sequence to pair directly with the mRNA's codon, delivering the exact amino acid required. These 3 types of RNA work in unison to transform the copied instructions into a physical, functioning protein. This entire system relies on transcription to act as the ultimate safeguard, an elegant, highly regulated supply chain that ensures the silent secrets of DNA can safely become the active machinery of life.